Myself, Dr. R.Shunmuga, Vadivu, Assistant Professor, Department of Biochemistry, SRM Dental College. Have a nice day. Today we are going to discuss about the plasma proteins and immunoglobulins. Plasma proteins and immunoglobulins. Plasma proteins are essential biomolecules present in blood plasma. This lecture introduces the classifications, functions, and clinical importance of plasma proteins and immunoglobulin. Plasma is the non-cellular liquid portion of blood. It contains about 90 to 92% water and 8 to 10% solids. Plasma acts as a transport medium for nutrients, hormones, waste products, and proteins. Proteins constitute about 7 to 8% of plasma solids. The total plasma protein concentration ranges from 6 to 8 gram per dl. The major plasma proteins are albumin, globulin, and fibrinogen. Here, plasma proteins are separated by electrophoretic techniques, which separates proteins based on their electrical charge and molecular size. This technique is useful in diagnosing liver, kidney, and immune disorders. Here, general properties of plasma proteins. Most plasma proteins are synthesized in the liver except immunoglobulins. Most are glycoproteins except albumin. Each protein has a definite lifespan. For example, albumin has a lifespan of about 20 days. Plasma proteins, main functions of plasma proteins helps maintain osmotic blood pressure, regulate blood viscosity and pH, provide immunity, participate in blood coagulation, and transport various substances in the blood. Here the picture shows how the serum are separated, how the plasma are separated. Serum is equal to the plasma without clotting factors. This one is on MCQs. Plasma albumin performs the following function except maintenance of osmotic pressure, buffering action, transport of substances, clotting of blood. Now you have to find out the answer. Next, coming to this albumin introductions. Albumin is the most abundant plasma protein, forming about 60% of total plasma proteins. Its normal concentration is 3.5 to 5.5 gram per dl, and it is synthesized in the liver. Here, main functions of albumin are albumin maintains oncotic pressure, regulates fluid distributions, transport fatty acids, hormones, bilirubin, drugs, and minerals, acts as a protein reserve, and helps maintaining acid-base balance. Next, coming to the globulin. Globulins are larger than albumin and have a concentration of 2.5 to 3.5 gram per dl. They are classified into alpha 1, alpha 2, beta, and gamma globulins. Alpha 1 globulins. Alpha 1 globulins include alpha 1 antitrypsin, which inhibits proteases, thyroxine binding globulin, which transports thyroid hormones, and retinol binding protein, which transports vitamin A. Alpha 2 globulin include ceruloplasmin for copper transport, haptoglobulin, which binds free hemoglobin, and erythropoietin, which stimulates the red blood cell production. Next, beta globulins. Beta globulins include transferrin for ion transport, beta lipoproteins for lipid transport, hemopexin for heme transport, and prothrombin, which participates in blood coagulations. Gamma globulins. Gamma globulins consist of immunoglobulins that function as antibodies and provide humoral immunity. Next, coming to the last one, fibrinogen. It is a filamentous plasma protein synthesized in the liver with a normal concentration of 0.2 to zero point 0.4 gram per ml. It is converted to fibrin during blood clot formation. Immunoglobulins or antibodies. Immunoglobulins. Immunoglobulins are classified based on heavy chains. All immunoglobulins contain two heavy and two light chains. Immunoglobulins are glycoprotein molecules that are produced by plasma cells in response to immunogens which acts as an antibodies. There are five classes of immunoglobulin: IgG, IgA, IgM, IgD, and Ig. These are classified based on their heavy chains. All immunoglobulin contain two heavy chains and two light chains. Clinical significance. Alterations in plasma proteins levels indicate liver diseases, kidney disorders, malnutrition, infections, and immune-related diseases. Here, that immunoglobulins Ig is the only antibody for placental transfer of immunity. IgM is the most potent agglutinating antibodies. IgA is a mucosal barrier. IgE is main one for that antibody for allergies. IgD and IgM are present on the surface of. Here are the basic structures of immunoglobulin. Immunoglobulin have the heavy and light chains and disulfide bonds, variable and constant regions, hinge regions, domains, and saccharides. This structure shows that V and C regions, variable chain and constant regions, two chains, light chains and heavy chains. Most heavy chains contains one variable heavy chain and three constant domains: constant heavy chain 1, constant heavy chain 2, and constant heavy chain 3. Here the light chains may be either of two types, kappa and lambda, but not both one. Heavy chains may be of five types. So depending on the heavy chains only, they are classified at immunoglobulin as alpha gives that A, gamma gives that G, delta gives that D, and mu simple gives that M, and epsilon gives that E. IgGs are named as per their heavy chain types. Here are some of the immunoglobulin MCQs. The immunoglobulins are classified on the basis of light chains or heavy chains, carbohydrate content, or disulfide bonds. Know that it is heavy chains. All immunoglobulin contains 4 light chains, 4 heavy chains, 2 light chains, and 2 heavy chains and 2 light chains. Here you know that 2 heavy chains, light chains. Structure of IgG. Immunoglobulin contain a minimum of 2 light chains and 2 heavy chains. 4 chains are linked by the disulfide bonds. Here that antigen binding site is variable light chains and variable heavy chains, and thus biological activity occurs in the constant light chains and heavy chains. Each Ig molecule has a hinge regions between CH1 and CH2. Hinge region allows a better fit for the antigen surface. Structure of Ig domains. Ig is folded into globular regions, each of which contains an intrachain disulfide bond. These regions are called as variable light chains and variable heavy chains form antigen binding site. 3 variable amino acid sequences at the amino terminal end called hypervariable region. That is complementary determining regions. Specificity of these antibodies is due to hypervariable regions. So the capacity of this antibodies binding with the antigen determines by the hypervariable. Structure of IgG. Some of this Ig have oligosaccharides, that is, the carbohydrates are attached to the constant heavy chains 2 domain in most immunoglobulins. However, in some cases, carbohydrates may also be attached at other locations. Here, this enzyme, when immunoglobulin treated with the enzyme papain, digestion splits the immunoglobulin into two fragments. One is FAB and another one is FC. FAB is the fragment for antigen binding, which has the valence 1, specificity determined by the variable heavy chains and variable light chains. FC is crystallizable fragment, or otherwise called as fragment for complement binding, which has an effector function. Valency of antibody. Valency of this immunoglobulin refers to the number of antigenic determinants that an individual antibody molecule can bind. The valency of all antibodies is at least 2 in some instance. Functions of immunoglobulin. Some immunoglobulins also bind to receptors on placental tropoplasts, which results in transfer of the immunoglobulin across the placenta. As a result, the transferred maternal antibodies provide immunity to the fetus, and here we have immunoglobulin classes which can be divided into 5 classes. We already discussed that 5 classes. Classification depends on this heavy chains, gamma, mu, alpha, delta, and epsilon. Immunoglobulin G, a major immunoglobulin serum is immunoglobulin G. Major immunoglobulin in extravascular spaces is immunoglobulin G. Only immunoglobulin that crosses placenta is the immunoglobulin G. Here, this immunoglobulin G involved in this complement fixation, opsonization. Here, what is opsonization? Opsonin is used to describe the substances which enhances phagocytosis, that the antigens are engulfed by the cells. IgG is the good opsonin. The antibody prepares the antigens for killing by the phagocytic cells. Here macrophages, monocytes and neutrophils and some lymphocytes have Fc receptors for the Fc region of immunoglobulin G. A consequence of binding to the Fc receptors on such cells is that cells can now internalize the antigens better. Monomer, IgG is a monomer, major class of serum, that is 70% of total immunoglobulin. From the total immunoglobulin, it is IgG. Main antibody in the secondary response. Constitutes important defense against bacteria and viruses. Only antibodies that crosses the placenta. It's a maternal antibody that protects IgA, second most common serum immunoglobulin. IgA is the major class of immunoglobulin in secretions like tears, saliva, colostrum, mucus. Since it is found in secretions, secretory IgA is important in mucosal immunity. Normally, IgA does not fix complement fixations unless aggregated. IgA can bind to some cells, polymorphonuclear cells and some lymphocytes. IgA is a monomer, sometimes maybe a dimer. IgA occurs in two forms: secretory IgA, which is a dimer form, serum IgA, which is a monomer form. Second most abundant class of serum immunoglobulin, which covers mostly 20%. Major component of colostrum. This IgA present in saliva, mainly present in saliva, tears, and respiratory, intestinal, and genital secretions. This IgA protects mucous membrane from antigenic attack. IgM. IgM is the largest one when compared to the other one. It's a monomer or sometimes pentamer. 8 to 10% of normal serum immunoglobulins are IgM. These are produced early in the primary response to an antigens. Activate complement, promotes phagocytosis and causes lysis of antigenic cells. IgG is the monomer, less than 1% of serum Ig. No known antibody functions for this IgD. May function as an antigen receptors. IgD is an IgE exists as monomer. Extra domain, they have that CH4. Trace amount in serum, 0.004% is present in our serum. Involved in allergic reactions, binds very tightly with the basophils and mast cells, which is involved in. As a consequence of its binding to basophils and mast cells, IgE is involved in allergic reactions. Binding of the allergen to the IgE on the cells results in the release of various pharmacological mediators that result in allergic. Here, clinical importance of immunoglobulin. When there is an increased level of IgE, immunoglobulin, it causes multiple myeloma, liver diseases, and rheumatoid arthritis. If there is a decreased level of normal immunoglobulin, it causes A gamma globulinemia, HIV, and nephrotics. Antibodies allergic response. That's all about immunoglobulin. Try to answer for this MCQs. Thank you.